starting to write your protocol. So let me just forward the slides here. So we obviously can't cover everything in the hour that we have. So what Sharon and I did is we tried to identify some of the concepts that we thought would be the most valuable for you guys to start thinking about before we meet face-to-face in Ann Arbor. So we're going to touch on a series of things. We're probably going to move pretty quickly. We encourage you to ask questions in that chat box. I see that you guys are already using that well, so thank you. Ask us questions as we go. There may be things you're thinking about that we don't talk about, and you're welcome to bring those up at the end or in the middle as we talk. So let me tell you a little bit about what we're going to try to cover in the hour that we have. So we're going to talk about kind of an overview of a series of things. Sharon's going to talk a little bit about some of the concepts of design, which probably many of you are pretty familiar with. Then she's going to spend a fair amount of time talking about confounding and blinding. So these are concepts that we really have to make sure you have under your belt as we talk about developing your protocol. Then I'm going to talk a little bit about your target population and how you think about eligibility criteria, and then how you start thinking about how you're actually going to recruit your patients. And then we'll talk a little bit about your intervention and some of the kind of oddball things that you have to think about with that. And then Sharon's going to finish by talking about outcomes and some statistical analysis concepts and sample size. So we'll jump back and forth a little bit. I'll warn you that there are going to be some quiz questions in the middle if we can get that to work well. So stay on your toes, and I'm going to pass it over to Sharon to start with some of the concepts of trial design. So Sharon, do you want to take it?
Sure. Can everybody hear me? Okay, well, if you can't hear me, use the chat box to let me know. Okay, so I'd like to start today by talking to you about the phases of clinical research, and we're going to talk a little bit about the objectives phase by phase. While I don't think it's really necessary that every one of you has a specific label for your clinical trial, that it has to be labeled as phase 1, 2, 3, or 4. I think it's helpful to understand where in the development process each of your questions falls. And the reason I think that is because it will help you to keep in mind which things you really need to be focusing on. So in early phases, you might be focused on very different aspects of some of the topics that we're going to talk about today, and other aspects in later phases. And we'll talk about how these things sort of work together. So when I refer to phase 1, many folks refer to phase 1 as the first time that a therapy is tested in humans. Now, this is often the case when you're talking about a new compound, but in many of your studies, it might actually be the case that it's not the first time your compound is tested in humans, but it might be the first time that your compound is tested in a particular disease population. And from my perspective, that would still be considered a phase 1 study. The objective here is to test this intervention in a small number of patients with the goal of determining a safe dosage range, or perhaps identifying one safe dose, and to evaluate the safety profile, how tolerable your patient population finds it, and to identify any of those side effects which you're going to want to pay more attention to as your intervention continues to move through the process. Now, once you've identified a safe dosage range, or again, a safe dose to move forward, the next phase of clinical research is generally geared toward obtaining preliminary evidence of efficacy. The idea here is that you want to be able to get some sense of the signal, something that tells you that it's worth then moving this intervention even further in the development process. And finally, once you have established a safe dose, once you've tested it in a slightly larger population of patients so that you have a sense as to the side effect profile, so that you have that efficacy signal, you want to do a large-scale clinical trial in order to confirm the effectiveness in your treatment population. And most folks would consider that to be phase 3. Again, I don't think it's necessary that every trial is labeled with one of these phases. In fact, in some areas of intervention, we don't use this terminology at all, but it helps you to think through which key components of the design or of the protocol you really need to focus on. So let's look at each of these individually. In phase 1, these are generally dose-finding designs. And I have the term dose in quotes here because I'm using it very loosely. In the type of work that I do, dose is usually refers to a dose. It's a dose of a compound that I'm going to be administering. But it doesn't have to be a drug that we're talking about. It might be, dose might refer to the number of times that a treatment has to be received in order to elicit a response. Or it might be the duration of rehabilitation that's required to elicit a response. So dose can mean any number of things, and the same sorts of designs are applicable in many of these cases. These dose-finding designs are often used to estimate the maximum tolerated dose, the minimum effective dose, or some combination of these. I'll refer to it as the optimal dose. And you would tailor the design a little bit depending on which dose specifically you're trying to identify. But the definitions of these are going to be very specific, okay? It's very unlikely that even if 3 of you have the same dose-finding sort of question, it's unlikely that you're going to define the maximum tolerated dose in exactly the same way. And that's one of the things that you need to think through as you're writing your protocol if you're in this dose-finding space. What does the maximum tolerated dose mean for your specific intervention? We recently completed a study of deferoxamine and ICH, and for us, the maximum tolerated dose was defined as the dose at which no more than 40% of our patients experienced a dose-limiting toxicity. Okay, so that 40% number, that's something that was chosen by the clinical team after reviewing the literature and discussing what exactly we meant by a dose-limiting toxicity, because that's going to be another question that you have to address in this space. What specific adverse events would make you want to consider that this intervention is not working for your patient population, that it's too toxic, okay? It might be any serious adverse event that occurs you might consider to be a dose-limiting toxicity. Or you might consider any serious adverse event that occurs during the acute treatment phase. Or even more limiting, any serious adverse event that occurs during the acute treatment phase that can be at least possibly related to the intervention itself. Okay, so there's some leeway in how you're making these definitions, and again, it's going to depend on what the literature suggests, both for your patient population and for the intervention that you're studying. Okay, the same would be true for the minimum effective dose. You might define the minimum effective dose as the dose at which at least 50% of your patients achieve a favorable outcome. But that definition of favorable outcome is going to be specific to your particular disease, your particular patient population. The rate that you're looking for, that 50%, is going to be specific to your patient population based on what proportion of untreated patients would achieve a favorable outcome. So again, you have to sort of think through these logistics as far as safety and tolerability goes. It's not sufficient to just say, I want to show that this particular intervention is safe. You have to think about what that means. There are 2 broad classes of dose-finding designs. I'm going to sort of lump everything in and say it's either an algorithmic design or a model-based design. The algorithmic designs, the most common is the 3+3, but there is a more general class referred to as the A+B designs. These designs are nice in that it can be very clearly, the algorithm can be very clearly specified in a flow chart that can be decided on at the beginning of the study, and you just follow the flow chart. As you enroll patients, you observe whether or not they have a dose-limiting toxicity event, and you just follow the flow chart to decide what to do next. The problem with those designs is that it's not very statistically efficient, okay? In the algorithmic designs, the decision to escalate to the next higher dose or to de-escalate to a lower dose are determined solely based on what is occurring in the currently treated cohort. It ignores all of the other accumulated data, and that's obviously not ideal, right? So the model-based designs actually fit a dose-response curve after each cohort of patients has been observed. And so the dose at which the next cohort will be treated is determined by that model. So that's very nice. It's very statistically efficient. It uses all of the accumulated data, and it's been shown to have really nice properties. There's been a ton of research in this area. But it is also statistically cumbersome, both on the front end and in the implementation, because someone has to be ready to update the model at the end of every cohort. Okay, and depending on how quickly your patient population enrolls or how quickly you need to treat them once you find the patient, that could be problematic. We did this for, again, the deferoxamine and ICH trial, and this was a couple of years ago before everyone and their brother had smartphones. I received a text message every time a subject was enrolled and every time a dose-limiting toxicity was observed, so that I could go in and update the dose response curve and let sites know what the next dose for treatment was. So it does require some additional statistical effort, but again, it makes better use of the data than the algorithmic designs. If you're in this space, there's a really nice paper, this Garrett-Mayer reference. Liz Garrett-Mayer is one of my colleagues at MUSC, and her paper in clinical trials actually describes the algorithm for the 3+3 design and gives a really nice tutorial on the continual reassessment method. So if you're in this space, I highly suggest taking a look at this paper. Once you've established a safe dosage range or a safe dose, and you're looking for that signal of efficacy, you've moved into the phase 2 space, or at least that's how I'm going to think of it. And the phase 2 space is, I think, the hardest space to classify, because there's a huge variety of phase 2 designs based on the question that you need to answer. And they're very specific, and there's a lot more flexibility in terms of how you set up the design, in terms of getting at the answer to the question. So it's both a nice place to be and a more complicated place to be. Probably the simplest phase 2 design is a single-arm study of your new therapy, and the results of that new therapy are then compared to the data obtained from a historical control. Okay, there are problems with comparing a new therapy to a historical control. If care practices have changed over the last however many years, if the measurement of the outcome has changed in recent years, then the historical control data you have access to may no longer be relevant for the current patient population, and that might lead you to make incorrect conclusions. But there's a huge sample size savings that can be achieved if you can make use of historical control data, either on its own or with a calibration control cohort. And so that's something to think about if you're in this space. A phase 2 design might also look like a multi-arm randomized trial. You might randomize patients to either the new therapy or the concurrent control and attempt to compare them at the end. Or you might randomize patients to multiple therapies and attempt to just select that one which looks the most promising. Okay, again, depending on what the objective you have in your phase of research, it may not be necessary to do a head-to-head comparison against a placebo in a phase 2. The outcome could be, as I said, selection of the most promising endpoint, which does not have to be based on a statistical hypothesis test. It might be a superiority test, but based on an established biomarker or some surrogate endpoint rather than the clinically relevant endpoint you might use in a definitive trial. Or it might be a futility test based on a clinically relevant endpoint. And this last bullet here is just to remind me to tell you that in this space there's a lot of possible adaptations. I can't even begin to talk about them all, but I want to say that with the caveat that you have to have a very clear idea of what the question is that you want to adapt to. Because any adaptation throughout the stages of clinical research are supposed to be pre-specified in advance. That means when you're writing the grant, when you're writing the protocol, when you're writing the statistical analysis plan, it should be very clear, if this happens, this is how we're going to react to it. Okay, so we're not adapting on the fly. We're adapting after very careful consideration of what the possible outcomes are and what the possible next steps should then be. And at the bottom of this slide, I provided you with a reference. This paper in ALS, again, about 10 years ago, is a very nice example of how these different possibilities that are on this slide can be merged together to answer the question of interest. So this particular group of investigators was looking at coenzyme Q10 in ALS, and they had 2 doses under consideration, and they wanted to know which one was best and was it worth testing in a confirmatory trial. And so what they did was they set up a 2-stage clinical trial where in the first stage they randomized to 2 separate doses and to a placebo. And at the end of that first stage, they selected the dose which looked the best. And then in stage 2, they continued randomizing to the selected dose and to the placebo. And at the end of the whole trial, then they did a futility test to decide whether or not it was worth moving the selected dose forward to confirmatory testing. Again, it's a really nice example of how these designs can be tailored to answer the question that you're trying to ask, while of course trying to maintain the statistical validity that we need to see from all of our stages of clinical research. And the phase 3 designs, I'm not going to talk too much about. Many of you are familiar with them just from reading the literature. Many of them look very similar. It's often a comparison of new therapy to a current standard, just a 2-arm clinical trial. The outcome is typically a clinically relevant endpoint, although how we look at that endpoint depends on the question that we're asking. So it might be a superiority test. For example, if you're familiar with the PROTECT clinical trial, we were testing the superiority of progesterone over placebo in traumatic brain injury patients. In some cases, you're not interested in testing superiority. You may not even expect that your new intervention is superior, but it might have other advantages that would make you want to consider it. The Rampart trial, which was completed a couple of years ago, is a nice example of a non-inferiority study. Or you might be looking at establishing equivalence of 2 interventions. These phase 3 designs often include interim analysis for efficacy and or futility. And the idea here is that phase 3 trials are very resource intensive. They enroll either hundreds or maybe even thousands of patients across tens or hundreds of sites. And it requires a lot of effort, both to keep the trial up and running and to obtain the data that comes in at the end. If at some point during the accumulation of data, we can see that the new intervention is overwhelmingly efficacious, then it's an ethical problem for us to continue randomizing patients to the substandard intervention. And so that interim analysis for efficacy allows us to terminate early in the face of such data. On the other hand, if as we're accumulating data, it looks like we are never going to be able to demonstrate that our intervention is efficacious, then it's a waste of resources for us to continue randomizing patients when we're never going to get to that answer. And again, here, the interim analysis for futility would allow us then to adapt to that information. Those are referred to as group sequential designs, and in fact, those are adaptive, although they're not the exciting version of adaptive that we're used to thinking about. Adaptations in the phase 3 space often look more like adaptive randomization, either adapting to the covariates or adapting to the responses that you're observing, or perhaps sample size re-estimation to adjust nuisance parameter estimates, things like that. Okay, so once you have considered which space of clinical research you're in, the next thing you need to think about is what other variables are there that could impact my outcome, right? Because at the end of the day, you want to make sure that you have a solid answer to the question that you're asking. And so you need to think about what else could impact the results that you see. And so specifically here, I'm going to focus on confounding. The idea is that a confounder is something that could potentially explain the results of your clinical trial. And the real concern is that the treatment arms might differ with respect to a known prognostic factor. Okay? If that happens, the treatment effect might be tangled up with the effect of that prognostic variable, and it could distort the truth that you see. So again, I'll give an example. If you're familiar with the NINDS-CTMC study, there has been a number of papers over the year which have suggested problems with balance in terms of age or the NIH stroke scale score, which represents severity in this patient population. So if you conduct a clinical trial like the TPA study and NIH stroke scale score turns out to be imbalanced across your treatment arms, so that perhaps the treatment arm has milder injuries than the placebo arm, but you ignore that information and you go on to do your analysis at the end of the trial and you show that treatment looks better, it had better outcomes under the intervention. The question there is, did the treatment really impact outcomes and make them better? Or did the treatment arm look better because they started out better, right? And if the imbalance is too drastic and you don't account for that somehow, you can't disentangle those two effects. And this is essentially the problem with confounding. So a true confounder must be associated with the outcome, it must be associated with the treatment, and it can't be in the causal pathway between treatment and outcome. Okay, so while you're doing the study, you're thinking about potential confounders, right? Because you don't know if it's going to be associated with treatment, but you can address the other two during the design stage. And again, the potential impact of confounding is one of two things. Either the confounding could disguise the true treatment effect, meaning you don't see something that should be there, or it could create an apparent treatment effect. And either of those cases, of course, is a problem, and we're going to want to try to address those either statistically by addressing potential confounders in our randomization scheme, or putting potential confounders in our analysis model, or logistically, which Karen will talk about in a little bit. And again, I've given you a reference for a nice paper on the importance of confounding in clinical research. Okay. The last thing I'm going to talk about before handing it over to Karen is this issue of blinding or masking. So in confounding, we're worried that the presence or the imbalance of this known prognostic variable is going to distort what we see. Blinding and masking is another place where what we should see can be distorted. Bias is referred to as a systematic effect, okay? Blinding and masking is used to control the bias that can come from either the investigator or the subject knowing which treatment they received. And this is pretty easy to think about, right? In an unblinded study, both the investigator and the subject know what they received. Okay. Sorry, Robin just asked if there's a difference between blinding and masking. So I'm using the terms interchangeably here. The idea, again, is that you want to keep either the patient or the investigator, ideally both, unaware or blinded to or masked from the treatment that they're receiving. So in an unblinded study, especially if the response is subjective, okay, so the investigator knows what the patient is receiving and the subject knows what the patient is receiving. The subject might say, well, I know that I'm receiving the control, so I'm not going to, I can tell I'm not doing as well as I would be doing otherwise. And so their response information back to you might reflect that information. In a single blind case, the investigator knows, but the subject does not. And this issue of bias is still present. It's just that it might manifest in a different way. So in this case, if my doctor knows that I was randomized to the control arm, he might prescribe different concomitant medications, maybe to help with my symptoms, maybe to help get me to the outcome he thinks I should achieve. And that can impact your assessment as well, because it's not going to be balanced across the treatment arms, right? It's going to occur more in one than in the other. In the double-blind case, which is typically the gold standard, neither the subject nor the investigator knows. This is not always possible, but it's often what we target. And there is a third, actually, which most folks don't think about, but the triple-blind case means that neither the subject nor the investigator nor the Data and Safety Monitoring Board knows. In practice, this is hard to pull off. for very long, right? The Data and Safety Monitoring Board is going to take its responsibility toward patient safety very seriously. And if there is any evidence of even a potential safety concern, they have the right to request that they become unblinded so that they know which arm is which. And in that case, the triple blind then reverts back to a double blind. Okay, so I'm going to wrap up this section with a question and then turn it over to Karen to talk more about the trial logistics.
Good, so hopefully everybody's choosing their answer. You'll all be able to see the question on the screen. We only have 10 people who have voted, 11 people who have voted. So we have about 25 of you that still need to vote. All right. So we have about 22 votes in. Sharon, do you want to comment on this? It looks like the group did really well.
Yeah, that's great. So yes, a true confounder must not be in the causal pathway. If a potential confounding variable is not associated with the outcome, then it doesn't matter whether it's balanced or imbalanced. It's not having any impact on your trial results. If it's not associated with the treatment, then the impact on your treatment effect estimate is minimum. And it should not be in the causal pathway between treatment and outcome. I see there's one more question in the chat box on combined phase 2-3 trials. We could do an entire course on adaptive sequential phase 2-3 designs. There are possible. There are lots of great texts on them, but I'm not prepared to talk too much about them today.
Good. Okay. Nice job on that, you guys. We're going to keep going. We're going to talk a little bit about your patient population now. We're going to start conceptually with just thinking a little bit about your target population. So all of you should be thinking about who's ideally in your target population. So for a stroke trial, it might be all ischemic stroke patients within a 12-hour window are the target. Or for a GBM trial, all GBMs, regardless of whether they're new GBMs or recurrent GBMs, that they're all in your target. Or for an MS trial, it might be a population of MS patients who have failed a certain number of previous medications, something like that. So think about who your target is. Who would you like to capture in your trial so that you have presumably a new treatment for that population? And then what we'll do is we'll start to consider the issues that come along with thinking about your target population. So then we'll have to think about, is it feasible to get all of those people? Is it going to be confounded? Some of the concepts that Sharon just talked about. Is it going to be generalizable? We'll dance with that quite a lot in terms of when do we give up generalizability, when do we fight for generalizability, and then we'll think a little bit about issues that relate to actually being able to recruit this broad population that is your conceptual target population. Okay, so with that, then we have to start thinking, once you understand who your target is, we want to start thinking about how you operationalize the eligibility criteria so that you are capturing as many of the target population as you would like to capture. So ideally, your eligibility would exactly match your target. What we know is that that rarely happens. The first thing for most of us, almost all of us, and especially you guys because you're doing comparative trials, is that there's this component of consent. So already, if we have a target population and 10% of them say they don't want to participate, then we've lost that population from our target. So almost all of you, if not all of you, will have that in part of your inclusion criteria. And then we'll think a lot about, again, these same concepts that you'll hear about as we continue to talk about feasibility, confounding, all those issues, and balancing those things with trying to capture the population that we're interested in. So let's think about inclusion first. So you're going to actually literally make a list of inclusion criteria that the patients who are being considered for your trial will have to meet before they will be offered an opportunity to become a subject in your trial, right? So you will literally list point by point what the inclusion criteria are. So again, ideally, we'd like it to be very close to your target group. So I'm a stroke guy, so for me, I'd love my inclusion criteria to be all acute ischemic stroke patients who can be treated within 12 hours. That would be beautiful. That's probably not likely, right? Because my second inclusion criteria, at minimum, is going to have to be that somebody provides informed consent. So again, most of you will put that eligibility criteria right there in the inclusion. Your grant writing committees who are reviewing your grants really, really like to see that you've thought about the fact that the subject or the subject's legally authorized representative has to give consent. So I strongly recommend that you all consider putting informed consent obtained in your inclusion. I recommend you not put it again in your exclusion criteria. That's just redundant, but I recommend that you all have it there in your inclusion. And then there may be other things. So there may be age or severity. Sharon talked a little bit about how severity is going to play a role for some of you. All those things will be in inclusion. That will generally be a pretty small list of bullets. Then we get to exclusions, and exclusions will generally be a much more challenging issue. So in general, we see a lot more exclusions as you start thinking about issues of confounding and feasibility and all the issues that we'll struggle with in terms of actually getting these types of patients into your trial as research subjects. So two of the things that we think about as we're developing our exclusion criteria will be balancing the issues of excluding confounding. So as Sharon talked about, you have these potential confounders, and one of your options is to say, you know, if the confound is, for example, renal failure, so I have renal failure on the slide, if that's potentially confounding in my disease, if you have renal failure or not, one of my options is to say, I'm just going to exclude everybody with renal failure. Then it's really clean, right? Then you don't have to worry about Sharon's issue that she brought up of whether there will be the same number of patients in one group as the other group that have renal failure, so that it's balanced. There are lots of other options, statistical options, but one of the logistic options that she was mentioning is you can actually exclude people that have a major potential confounder so that you don't have to deal with it. Now, in something like severity of your disease, you cannot exclude the severity of the disease, right? But what you can do is narrow it so only one type of severe patient, severe or non-severe patient, is included in your trial. Again, that may be problematic because we're balancing it with generalizability, right? So you want your trial, when it's over, to be generalizable so that you can share it with as many patients as possible and have an impact, right? So you'll always dance between that, yeah, it's confounding, but yeah, I want it to be generalizable. So think about that issue. The other issue is that you've got to balance your exclusions with issues of recruitment feasibility, and we're going to talk a little bit about recruitment, but you've got to make sure that you don't have 59 exclusions so that every patient that is considered to be a potential subject in your trial is excluded by one of those issues, and so then you really don't have a population that meets those criteria that you can enroll in your trial, and then you can't finish your trial. So those are important issues to think about too. The other thing I would encourage you all to do is to think about why you're excluding somebody. So if you're going to put a bullet point in your exclusion criteria, think about why you really need to exclude that group. And you should be able to actually have a concise, clear reason for excluding them. And as you write your grants, you will need to be able to explain very cleanly and crisply why you're excluding that population. So for children, for my disease, stroke, we almost universally exclude children in adult stroke trials. So it's very easy to write a statement saying this trial is for persons greater than or equal to 18 years of age. We have excluded people under the age of 18 because most strokes that occur under the age of 18 are a different disease with different causes and different outcomes, period. It's a different disease. That's why we exclude them. Not because it's easy, it's easier to exclude children, because there's a good scientific reason. They are fundamentally different. So hopefully that makes sense. Okay, so that's all I'm going to say about eligibility. Let's talk a little bit about how eligibility then blends into recruitment. So recruitment considerations are probably one of the most important things that you guys will think about as you start to develop these protocols and think about how to be successful in executing the protocols. So hopefully all of you recognize that recruitment is profoundly unpredictable. And though there are a small number of examples of trials that have had overly rapid recruitment, usually the issue is slow recruitment. And you can get into trouble with overly rapid recruitment. We can talk about that if we have time, but slow recruitment is much more commonly the issue, and that can get you into big trouble in terms of being successful in completing your trial. So I recommend that you all have a backup plan if your recruitment plan is not recruiting at the rate it was expected to. So for example, if you are planning on a single site trial or a 2-site trial, you should have a backup plan if those 1 or 2 sites cannot recruit enough subjects in the time frame, the early time frame that you're thinking about, and then be able to adjust so that you can get the full recruitment. And we can talk about some of those things when we're face-to-face in Ann Arbor. Startup, this is another thing that I commonly see in grants that come in, is that people forget that there's a huge investment that has to happen at the beginning of any trial, but especially a comparative trial, because there are usually 2 or more groups that are being compared, and so there's a lot of preparation. So if you have a 5-year grant and you go in and say, I am going to recruit patients for 5 years in this 5-year grant, you are not going to get the grant, because the people reviewing it know that it takes time to get ready. We generally think 6 to 12 months, depending on how complex your trial is, but at least 6 months of what's called startup to get your sites ready, to get your IRB done, all your regulatory issues, to get people trained, and to get your intervention prepared, whatever it is. And then you have a recruitment period, but then you have a follow-up period. So don't forget that you have to finish the last follow-up before you can start the analysis. So there should be a follow-up period and an analysis period. So it is not uncommon to have at least 1 year to 2 years of either startup or follow-up and analysis, so that your recruitment period is that time that's left in the middle. So make sure you think about that as you prepare your timelines. Exclusions, we've talked a little bit about this, but as you develop your plan for recruitment, I strongly recommend that you literally go through each of your exclusions and figure out what proportion of your population is going to be affected by that exclusion. So again, for me as a stroke guy, what proportion of the stroke patients that come to my institution actually get here after 12 hours? If my target population are stroke patients within 12 hours, then everybody who arrives here after 12 hours is absolutely not going to be considered eligible. So I need to calculate that in as I'm thinking about how many patients will be available. Also, remember, lots of patients don't want to consent, or they can't consent. In neurologic disease, there are lots of people who can't provide consent, and if someone else is not there to provide consent or assent, then those patients are not eligible. And then you have missed patients. There are always patients that somebody forgets to think about it, or people couldn't make it in to evaluate the patient, or something happened and the patient was not offered the opportunity. The other thing I want to mention quickly is selection bias. Some of you probably have bigger issues with this than others. Some of you may not have a big issue with this, but at least in some of our trials, there's an issue of selection bias. So is there something that is systematically resulting in us getting certain patients and excluding other patients? So the classic example, at least in stroke trials right now, is competing trials. So as you know, there are lots of stroke trials that are always ongoing. But if I'm at a site and there's a 3-hour window trial and a 6-hour window trial and then my 12-hour window trial, it would be a systematic selection bias if all the patients who got here super early went to the 3-hour trial, the folks kind of in between went to the 6-hour trial, and the only patients that came to my trial were after 6 hours and before 12 hours. That would be a systematic selection bias such that when I presented my final data and talked about my target population, the group that I recruited was not actually in my target. If my target was acute ischemic stroke patients up to 12 hours, because now I've carved out most of the 3-hour folks, most of the 6-hour folks, it no longer represents the group that I'm targeting. So that would be a systematic bias in who's in the trial. So make sure you're thinking about those things as potential obstacles. The last thing I'll say about recruitment considerations, and probably the most important message, if you remember nothing else about this slide, the total number of patients at a site with that condition does not equal your recruitment estimate. So if I have 600 ischemic stroke patients a year at my site, I should not come close to expecting 600 patients will be eligible for my ischemic stroke trial. You've got to think about all the exclusions and all the reasons why some of those patients will not fit in to your eligibility criteria. That's an important point when you're writing your grants and your proposals. Okay, I did want to say just 2 words about the intervention. Some of you will have bigger issues with the intervention than others. For those of you who are doing something behavioral or a therapy or something like that, it may be less of an issue. But for those of you who are thinking about your intervention to be either a medicine or a device or some kind of device-related treatment, some of these issues will be really important just to start thinking about. So regulatory, you're probably all very comfortable with IRB issues, which is great. Those are important. I get that most of you are comfortable with that. If there are issues with the FDA relating to an IND or an IDE, which are your intervention, your investigational new drug or device permissions, you've got to start thinking about those early. Those universally take a long time to get permission from the FDA to do experimental work with a new drug or a new device. So please make sure you're thinking about that if you think you may be affected by that. Blinding, Sharon talked a little bit about blinding, but remember there are lots of issues with blinding. So I get that you guys know that, you know, one pill has to look like the other pill, or the color of one thing has to be like the color of the other thing. But don't forget there are more subtle things. So just because you have 2 white pills doesn't mean that if you're telling the patient one is a sugar pill and one is not a sugar pill, and one tastes like sugar, that's a problem, right? That could be unblinding. Or if there's some kind of taste that they can be aware that whatever the interventional drug is, that it has a bitter taste or something. You've got to think about subtle things like that. Liquids in an IV infusion that stick to the tubing, that's one that can be unblinding to everybody in the room. The other thing to think about is you may, as Sharon talked about, have unblinded personnel. So somebody may be unblinded. The surgeon that does the surgery in one patient and does a sham surgery in the other is unblinded. So you've got to think about how you compartmentalize the fact that they have information that you don't want others to have. You don't want the patient to have it potentially, or you don't want the other investigators to have it. You have to think about how you're going to keep the other people blinded if your intent is to have some of those people blinded. And then access to the intervention. I won't talk much about this because we're short on time, but think about how are you going to get the drug? How are you going to get the control, the placebo for the drug? Do you have to make it? If you have to make it, that's a big deal. How are you going to get the device? How are you going to get the therapists who are going to do the intervention? Do you have the space you need for your infusion therapy? Whatever it is, think about what access you have to the things you need to operationalize actually offering this. And then I just want to say 2 seconds about calculating the volume. So most of us think about, the volume of our drug is I need 100 patients and every patient's going to get 1 pill. So I need 100 pills. And if you calculate your volume of what you need for your intervention that way, you're going to be sadly disappointed probably about a third of the way through the trial. So there are issues with shelf life, for one thing. Most of the drugs that are used for interventional trials have some sort of shelf life. They're only allowed to be distributed for some period of time, and then you have to restock. And again, the timeline of your recruitment affects this kind of thing, because if you plan to restock twice, but it takes twice as long and now you have to restock 4 times, that's much more drug that you had to buy, which affects your budget and your the timeline of making the drug and getting it on the shelf, things like that. Devices as well. Don't forget your devices, your computers. For our trial, we have computers. They have a shelf life. They will not last forever. And you have to calculate those things in as you're thinking about your protocol. And then replacement or dropouts and storage is the other thing. If you're going to get, 40,000 computers, then you better have a space to store 40,000 computers. I know I'm exaggerating a lot, but it makes the point. So that's all I'm going to say about the intervention. As we start to meet face-to-face, then we will have a better idea of what everyone needs in terms of their particular interventions. So we're going to move on to our next quiz question. So as you can all see on the screen, here's the question. When specifying eligibility criteria, one should consider which of the following? Is everyone able to vote? You should be able to click on your screen what your answer is to vote. We have about 27, 28, so I think we have most people. So can I broadcast this? There we go. So you guys did really well on that question. Indeed, the answer is all of the above. Each of these things is really important as you start thinking about your eligibility criteria. So we are very short on time. I'm going to move it to Sharon, and she's going to last. Sharon, are you there for the last few seconds?
Yes, I'm here. Okay, so we're going to do this very, very quickly. And in part, that's on purpose, because we could do a 2-semester course on the statistical approach and how to decide what it should be and how to compute the sample size. And so we're just going to sort of gloss through that. But what I do want to talk about for just a few minutes is how to choose your endpoint. I want you to think about all of the things that go into the outcome that you're going to use. Think about what could impact them and make that decision very, very carefully. We try to pre-specify what that primary outcome is going to be, and all of our decisions about how the trial should be set up, how many subjects we need, and things like that are tailored to that choice of an endpoint. So we can't just sort of willy-nilly move them around. So we really need careful thought and consideration into what endpoint is going to be used for a given clinical trial. I want you to think about, if you're choosing between endpoints, whether or not you have an objective versus a subjective endpoint. A subjective endpoint might be something like the modified Rankin scale, okay, where there's a little bit of variability between assessors. An objective endpoint would be something like mortality, where it's very clear either the patient is alive or they are not. Subjective endpoints have, especially in the case of the MRS, or the modified Rankin scale, can provide much more information, and they are often very useful endpoints. When you're using something like that, it's even more important to consider these other things, these other points. How much training or certification is going to be required for your outcome assessors? If you have a subjective endpoint, something where there is variability between assessors, what can you do to make sure that that variability is minimized? for the modified Rankin scale, there is a certification. For many other scales, there is at least training that can be provided so that patients, similar patients tend to be scored in the same way across assessors. And so you want to sort of standardize that as much as possible. You want to think about whether or not your particular outcome has to be done in person in the clinic or whether it can be obtained over the phone. Loss to follow-up is going to be something that will need to be considered. I'll talk about that in a minute. But loss to follow-up for the primary outcome can be minimized if you have an assessment that can be obtained over the phone when you can't get the patient into the clinic. If you have an imaging or a biomarker outcome, whether or not you need a central reading of that outcome or the local reading will suffice is an important consideration. A central read is going to be a lot more resource intensive than the local reading, but if there's enough variability across sites or across assessors in your outcome, then it might very well be worth the additional resources to get that central read accomplished. The timing of the assessment should be considered. If your primary outcome needs to be at 6 months versus at 3 months, of course, it's harder to follow patients for longer periods of time, but if you need that additional time in order to truly see the treatment effect of your intervention, then you want to make sure that that assessment is placed at the right time point. And you want to think about whether or not you have the option to use a continuous versus a binary outcome, all other things held equal. So if survival is your question, you can, of course, treat survival as a binary outcome. Was the patient alive or dead? And it's a very simple analysis. But the continuous analysis of the time to death might be more informative, might be more statistically powerful, and you might be able to get more bang for your buck that way. So again, just sort of thinking through these things when you're making the decision will help. As far as the statistics goes, the statistical approach is going to depend on everything that we've just talked about. The study design, the endpoint we chose, how much loss to follow-up we anticipate. In many of the later phases of clinical research, we're doing an intention to treat analysis, which means everyone gets randomized. Everyone gets analyzed to the treatment at which they were randomized. That means even if I'm missing an outcome, I have to include them. And so that's an important consideration because they can dilute my treatment effect. And whether or not it's necessary to adjust for those potential confounders will impact our analytic approach. And then, of course, the sample size depends on that approach. So in many cases, this is an iterative process. We make all of these decisions, we determine the analytic approach, we calculate the sample size, and then find out it's too much. It's more patients than we can come up with, and we have to go back to the beginning and start thinking through what alternative choices can we make that will still answer the question, but allow us to do it in a more feasible manner. Okay, so one more question, and then I think we can take some questions. Okay, so that looks like most everybody, and you're exactly correct. We need to think about all of those things. So the sample size is going to be determined sort of later on in the process of thinking through your protocol. Karen, should I turn it back to you to summarize and take questions?
Sure. Get my camera on here. Okay, so just in summary, guys, what we've tried to cover is just a little bit of the conceptual kind of starter thoughts about operationalizing your research question, things that we want you to be thinking about, including which design is best for me, or what are my questions about design that I need to think about? What's my population? What are the tricky things about my intervention? Which outcomes are best for my trial? And we'll talk a lot about this when we're face-to-face, but what should my statistical approach be? So you guys all know that there's a whole group of folks that are going to help you with all this. We certainly are here to help. Sharon and I have our emails here, so for anybody who doesn't get to ask a question right now, we want you to email us. We'd be happy to follow up with you by email. And then you can put your questions in the chat box. So far, there have been a couple questions about data safety monitoring boards. So remember that in many of the trials that are comparison trials, there has to be some safety oversight plan. And for a lot of the phase 3 and actually a lot of the middle phase trials, a formal DSMB may be that safety body. And so there were some questions about blinding that group. There is a constant discussion about whether that group should be blinded or not blinded. Remember, every time we're unblinded, there's the potential for us to be biased toward or against something. So the purists believe that the more blinded people are, the more objective they can be in their assessments. And one of the things that Robin Conwit wanted to point out was that some DSMBs, and most DSMBs to start with, are either blinded or what we call A-B unblinded. So they know one group is doing one thing, the other group is doing another thing, but you don't actually know which one got which. And that can be a nice intermediate so that you have an idea that there may be differences between the groups, but you're really not sure which group is doing what. So it looks like we have somebody typing. So Sharon, chime in if you want to comment on DSMBs as well. Okay, who sits on a DSMB? This is a great question. So I would say universally, there is a statistician on a DSMB and a primary disease content expert. So I would say those two are probably the standard. And then you have lots of people that have other areas of expertise that might be on the data safety monitoring board. So they could be a radiologist, they could be somebody in a different area of expertise. In our clinical trial, we are doing a stroke trial with primarily diabetic patients. So we have stroke people on the DSMB, but we also have an endocrinologist on the DSMB, so another content expert. But almost always there's somebody who's at least one, if not multiple statisticians and multiple content experts. Sharon, chime in if you have other things to say. Why do we have more than one person doing this task? Usually, so that the question again is about the DSMB, why do we have more than one person on a data safety monitoring board? The point is that the board together brings expertise and thinks about safety and I would say data quality and data relevance and all those things. We call them safety boards, but in truth they cover a broad range of important things that have to do with making sure the trial is successful. And they bring all that expertise and then they consider things together. Some of these decisions are really tricky to make and you count on your very smart colleagues to think it through with you and to bring an area of expertise that you individually might not have. So I have to say I'm a big fan of moderate-sized DSMBs, not the teeny tiny ones, because having a lot of smart people in the room helping to make sure that your trial is on track is, in my opinion, always a good thing. So we have another question. Are there times when you would have an interim stopping criteria for a middle phase, a phase 2 trial? Sharon, I'm going to pass this one to you, but again, I would say this is a great question.
So I think the answer is yes, although you would have to think through what those stopping criteria would be. You might have safety stopping criteria. So if you know that, for instance, in an untreated population, the rate of a particular safety event is X, and then you might put in place stopping criteria that say at the first sign that the rate in my treated population has exceeded this X, I'm going to stop. You could also think about futility criteria, I think, if depending on how your phase 2 is designed. So if you're doing a superiority design in a phase 2, looking at a biomarker or a surrogate endpoint, you might put in a futility boundary that says if it's unlikely that I'm going to be able to show that my intervention is better on this biomarker, then I'm going to stop. So I think it's possible. I'm not sure how common it is. I know that we've talked about it In the futility space, we've talked about a futility stopping criteria, but in that case, it actually doesn't make a whole lot of sense. But I think depending on what you want to stop for, it could be considered, sure.
And Joe, I would add one other thing, is that feasibility. So again, if you create a trial and you say you're going to enroll stroke patients within 15 minutes of their stroke onset, and you need 200 patients in your middle phase trial to demonstrate that this is feasible, and 3 years into your trial you've not enrolled a single person, we have a feasibility issue, right? So you can actually have some kind of feasibility assessment somewhere down the line, and maybe they have a little less formality to them, but I would say that probably feasibility in the middle phase trials is one of the biggest things that we have to think about, because if it's not feasible to do what you're thinking about doing, then that's a really good reason to stop and rethink. So the next question, Sharon, I'm going to give this one to you also, is investigating rare diseases. So when you have a rare disease, from a methodological standpoint, how can you deal with sample size issues when you have a small total population, not just target population, but total population? So Sharon, do you want to talk about design issues that you can manage only having a very small sample available?
Yeah, so that's a really good question, and it's a really hard one to answer. I think if you are dealing with a rare disease, you want to be really, really efficient in how you're trying to go about answering your questions. And so you want to look for designs that are going to make the most efficient use of the data that you have. So things like the continual reassessment method, where you are making efficient use of all of the data that you've accumulated. You might be considering putting together adaptive sequential design so that the same patients who are being used maybe to select a dose can then be used to assess for an efficacy signal, or maybe the same patients who are being used to get at that efficacy signal can then be used to confirm efficacy. I'm not by any stretch of the imagination an expert in rare diseases, so there may be other things, but I think that you more so than others would want to be really efficient and really creative in how you put those designs together to make sure that you're getting every bit of information from those patients that you can get.
And Sharon, I think you mentioned earlier the historical control, especially in middle phase, may be a very valuable way to save kind of the impact of your intervention on the patient population, and sometimes that will work.
Absolutely.
Okay, so it's time after 1. We are over time. We thank all of you for joining. Will, I assume you still can't speak to the group, so I'm going to close for all of us and say thank you everyone for joining. Again, our email addresses are up there on the screen. If there were questions you wanted to ask and didn't get a chance to, we encourage you to send us an email. So thanks again for joining.